fungi. So yeah, perfect.
Hi, many thanks for the invitation, and I'm happy to present. Just trying to make it full screen. Can you hear me? Yeah.
Yep.
I will present the work we have been doing in the team about the domestication of Penicillium cheese fungi. You can see here the Penicillium roqueforti that is used to make blue cheeses, and here the conidia with the spores of the fungus, and here the Penicillium camemberti used to mature the camembert cheese. That the crust, you can see it's a Penicillium mold, and you can see here the Penicillium roqueforti and camemberti on a petri dish. So they are really mold that we use to transform the milk into a good cheese. And so, as you all know, domestication has been used since Charles Darwin time to understand evolution and adaptation. So he used pigeons, for instance, to understand how the diversification of life occurred and how adaptation occurs, and using really artificial selection as a model for understanding natural selection. So there's two components in Darwin's theory that we can try to apply to our mold and domestication in general. Is really the within population adaptation that needs, as you know, genetic diversity to occur. Natural selection, so is like you sort among diversity the most performant alleles, and also the diversification, so how you can generate different varieties from a common ancestor. Plants have been studied a lot for domestication. Like we selected the teosinte, the ancestor of the maize, to make corn, so increasing yield and the size of the stalk and the shape of the plant. So you can see here the picture of the ancestor of the corn and the corn, so with drastic changes. And so that's adaptation. It's really a drastic change within population. But there have also been a period of diversification. So there have been early domestication with increase in size of the grain, and then really making a lot of variety in different regions and regions of the world. And more recently, much stronger selection towards unique varieties, and we plant a single variety all around the planet. And so you can see, for example, the variety of maize and corn breeds that we used to plant, to grow, and now we all know only almost the yellow one, the Leo big one. So there have been, during domestication, times where humans generated a lot of variety, diversification, and more recently, a much stronger selection for only a few highly performant variety. While we know that for future adaptation and improvement, we still need a lot of variety for adaptation to occur. And that one thing we know since Vavilov also, the Russian scientist who first studied the center of domestication of plants, that plants have been domesticated in different areas with local adaptation, and the most powerful way of improving variety was to have several regions where you had the selection for particular local conditions, and then sometimes seed exchange. So you have both different population, but also gene flow, and that's the most powerful way for adaptation and diversification. But we also know that domestication can have some drawbacks, especially when there is a too strong selection. So there is strong bottleneck, and there can be deleterious alleles that are dragged with the beneficial ones. Like for instance, in this dogs breed, they cannot reproduce by themselves anymore. Often they cannot breathe anymore. And in apples, for instance, we exerted a too strong selection so that, for instance, many resistant genes against diseases, like here the apple scab disease, have been lost and are only found in natural population or in closely related species. And also some disuse function with, for instance, the domesticated banana. So we much bigger and more nutrients, but at the same time, no more seeds, and so it cannot reproduce by itself anymore. So we study fungi, and so as I guess you all know, also fungi are very close to animals. They form the Opisthoconte lineage, so they have a lot of in common. And so we were interested in the penicillium. So penicillium is a huge genus, and so you have a lot of fruit mold, and also some soiled, some fungi we find in soil, but a lot of crop or food spoilers, and there have been several lineages that we have domesticated independently and sometimes for the same use. For instance, you have here the Penicillium camemberti and here the Penicillium roqueforti, and that are very, very distant phylogenetically. And so they are separated 30 million years ago. It's like humans from the spider monkey. So even if they are the same genus, they are really, really distant. And so we have independently domesticated these two molds to make cheese. And so that's a good model to understand whether there have been convergent adaptation and whether it was by the same genomic mechanism. So we can wonder what were the traits under selection for cheese fungi. For instance, for the Penicillium roqueforti, so used to make the blue cheeses, you can see here the spores forming the blue area in the cheese. So probably it was not intentional at the beginning. The mold came spontaneously, and they found maybe it was a good way to preserve milk. And then the humans begin to cultivate the fungus. You can see here we used to make bread, especially for that, burning outside to avoid any contaminants, and inoculating inside the mold to cultivate the spores. And so you could select the best cheese, cultivate its mold, and then reinoculate in the next cheeses. And of course, now we do that in petri dishes, and we can buy sterile spore solution to inoculate in the milk. So what trait can have been selected here? So maybe the blue, for instance, the blue color, probably the aroma and flavor developed by the cheese maturing the cheeses. Maybe the sugar, protein, and lipid metabolism to be more efficient at using these nutrients. Also, we know that a lot of penicillium fungi produce toxins that are harmful to mammals. So maybe we selected a strain that were not producing toxin, or maybe producing more toxin, because the toxin are actually like antibiotics to compete against other microorganisms, and maybe that could avoid some spoiler, unwanted mold from outside. And maybe we have selected also for salt tolerance, because precisely to avoid this spoiled cheeses, we salt a lot the cheeses. So first, we sequence some genomes and compare different penicillium species. And you can see here the Penicillium roqueforti genome outside, and then other Penicillium species, including camemberti. And the redder, the more similarities to Penicillium roqueforti. So you have a gradient according to phylogenetic distance. But intriguingly, what we found first was that you have like big chunk in the genomes that were either completely absent from the genome or completely identical across very distant species. So like I told you, it was like humans from spider monkey, but there were like a big region with hundreds of genes that were 100% identical and in different locations in the genome. And they were either completely identical or completely lacking from the genomes. So it was really like it was horizontal gene transfer having occurred very recently in human times. And we know in fungi that the hyphae can make anastomosis, so DNA transfer may be easier than other opisthocons. So it was like there was very recent horizontal gene transfer and only between cheese fungi. It was between Penicillium camemberti and Penicillium roqueforti, for instance. And precisely this horizontally gene region had some interesting genes like lactose permease, beta-lactosidase, so transforming the milk sugar, and other genes involved maybe in macroorganism competition. So it looks like this horizontal gene transfer were maybe adaptive. And very recently, it was discovered that by another team that these horizontal gene transfer were actually mediated by huge mobile elements that are called starship, and that have like a captain domain at the beginning and a cargo domain at the end, and they can move within the genome or between genomes or between species, and like they include a lot of genes and they can move genes between species. So then we focused on Penicillium roqueforti domestication history, and so we wonder whether the different blue cheeses were made by the same population, and also we know that silage, so the fermented plants that we give to feed cattle, they can be contaminated by spoiled by Penicillium roqueforti, and they are very toxic to cattle. And also there had some food spoiler are Penicillium roqueforti, like on the bread or fruits. And so we wonder whether these non-cheese population were maybe feral, so escaped from cheese, or maybe they were the ancestral-like population from which we domesticated the cheese population. So we sequence some strain from cheese on non-cheese population, and we found out actually that it was neither of these two cases. So we found two cheese population and one lumber food spoiler population and one silage population, and they were neither were nested in the other. So it was like they were independent lineage, so they were neither feral nor ancestral-like. And interestingly, so one of the cheese population was found only in Roquefort in France, like it's a protected designation of origin, and with some diversity. And all the other blue cheeses from everywhere else in the world was from another lineage and with no diversity at all, basically. It was like we kept in Roquefort, thanks to the PDO, a bit of diversity and ancestral-like population, while there have been a very strong selection from a single clonal lineage for all other blue cheeses worldwide. And only this lineage, the non-Roquefort one, had these horizontally transfer region. All the other population and none of these starship elements. And so the Roquefort is a small town in south of France where this PDO occur, and we have to use there for the PDO the local strain of Penicillium roqueforti. So the history of the domestication was like there would have been 2 independent domestication and a more recent one, and with very strong selection with a unique strain for all the non-Roquefort cheeses, and Roquefort population with a bit more diversity. And actually the silage and food spoiler population splitted much more recently, so maybe a recent adaptation to human resources, but unwanted one. And the horizontal gene transfer only occurred there. And then so we looked at the trait evolution of this population. And so for the production of toxin, indeed, actually the cheese population, they produce very little or not at all toxin. So it looks like there have been a selection against toxin production, and with some footprints of gene disruption in the biosynthetic pathways. We also found that the clonal lineage used for every blue cheeses worldwide actually grows better on cheese, but slower on poor medium. So it it has been a stronger selection than in Roquefort for metabolizing efficiently milk, but at the expense of being less well to grow on poor condition. We also look whether the Pensium Roqueforti could exclude competitor, maybe some food spoiler. And so we made bed of Pensium Roqueforti, and then we inoculated a challenger, a white challenger. And you can see again, the non-Roquefort lineage was better at preventing the other from growing. So again, footprints that there have been a stronger selection on this strain. And then we made experimental blue cheeses, and we found that the two cheese population made bluer cheeses than the silage or food spoiler population. You can see here much more blue in the cheeses. So it looks like indeed there have been selection to being bluer in cheeses. We also looked in the experimental cheese at the volatile compound that are typical of blue cheeses, aroma and flavor, especially the 2-heptanol compound. And we found that it's much more produced in the Roquefort population and a bit in the 2 cheese population, while the silage population didn't smell anything at all. And the lumber food spoiler smell more like a wet mob than any blue cheeses. So again, it looks like there have been a selection for making good cheeses with this population. And then we look at the ability to produce spore on the ancestral-like medium. We use the bread to make these spore powder to inoculate the cheeses. And this time it was the Roquefort population, so the ancestral-like domesticated population that produce more spore on bread, while the non-Roquefort one that had been selected more recently and is cultivated on Petri dish didn't show this pattern. And so the story then with the tray is like we have these 2 independent domestication events for the blue cheeses, and with a selection for bluer cheeses. I didn't show, but higher salt tolerance, faster growth on cheeses, on milk, and at the expense of slower growth under poor condition. And yeah, more diverse and appealing aroma. But they also have lost a lot of diversity and also fertility. So we tested their ability to produce the sexual structure that we can see here on Petri dish. We inoculated 2 different strains of opposite mating types, and at the border, at the encounter, they produce perithecia, the sexual structure with asci inside. But the cheese strain were very bad at producing this perithecia and the sexual spores. So they have lost their fertility because they are only cultivated asexually now. But so recently we discovered another cheese population. So it's in the French Alps, the Termignon blue cheeses that are produced only by 4 farms. And actually that the rare cheeses where the mold is not inoculated, it comes spontaneously from outside. You can see here the Termignon blue cheese and the mold colonizing from the crust. And actually, so these strains form a third cheese population with more diversity than anything else we knew from cheese population so far. And so that's very interesting to have more variability. And so we also look, but they had this starship, this cargo element. So it looks like there have been selection and domestication even in this population. And so that's a PCA with many traits, possibly under selection for domestication. And you can see here the non-Roquefort, Roquefort blue cheeses, and the silage and food spoiler. So the cheese and non-cheese population are very different, and the terminion looks a bit intermediate between the cheeses and the not cheese population for traits important for cheese making. So again, it looks like it's like an intermediate step of domestication. So, oops, sorry. The history would be, but the terminion strain are closer to the non-Roquefort population. So maybe it's like the ancestral-like population from which there have been a very strong selection giving the non-Roquefort lineage. And so now we're trying to cross this different population to generate variability for lipolysis, for instance, to be able to degrade the lipid in the milk, and for growth, for color, and for toxin production. And we could indeed generate some diversity and find QTL for some of the important traits for cheese making. And so more briefly, a bit on Penicillium camemberti time, so a very distant lineage and used to make the camembert and the brie. But so that are white now, but actually they used to be blue, like all Penicillium are. So you can see older painting from this date, and the brie was blue. And you can see here a picture also from 1953, where this brie is blue and this one is white. And actually, so the Penicillium commune from which the Penicillium camemberti has been domesticated, it was blue-green, and the Penicillium camemberti has been selected like an albinos individual, white, and growing more fluffy. You can see here it really grow more up than vertically. And it was really selected for being white because it looked less moldy. And again, so we sequenced some genomes from Penicillium camemberti, its closest relative Penicillium biforme, that is blue, and it found that we use it actually for making goat cheese and Saint-Nectaire and some other cheeses, and some dry cure meat too. And another wilder close relative that is more a food spoiler, really wild fungus. And interestingly, so again, the Penicillium camemberti had no diversity at all. It was just a clonal lineage, albinos clonal lineage, with a starship element, except one old strain we found in the National Museum of Natural History in Paris, collected in 1905, which did not have the starship element. So maybe the starship occurred after that. And the Penicillium biforme, so it has a bit more diversity. Some have the starship, other strains do not have them, but there are more variability. So again, it looks like protracted domestication with different steps and stronger and stronger selection and less diversity. And so the history is more like that, with Penicillium fuscoglocum, a lot of diversity, and the Penicillium biforme used to make goat cheese, still a bit of diversity, a bit of blue-green, and a selection for a white strain, and we almost no diversity, just a few SNPs, but still two varieties that have different color and a bit of different traits, but with very few SNP between them. And the same horizontal gene transfer as Penicillium roqueforti just before the selection. And so we also looked at different traits, like the white trait and the fluffy traits that we can summarize as colony opacity. And we found that an increasing fluffiness and white degrees from fuscoglocum to biforme to Penicillium camemberti, and less diversity within species. And we also look at the ability again to exclude competitor. And again, so we made either so no loan or Penicium biforme alone or Camembert tea, and you can see that the Penicium Camembert tea was very good at excluding Penicium Roquefort Challenger, while Penicillium biforme let it a bit grow. So again, it looks like there have been an increasing selection for avoiding food spoiler. We also looked at the ability to grow on cheese versus poor medium. And again, you can see that so the wild species has like an even ability to grow, while the Penicillium Camembert tea is much better at growing on cheese, but much poorer at growing on poor medium. So again, a selection to better grow on cheese, but at the expense of losing the ability to grow when there is little nutrients. And also, again, for the toxin production, we found an increasing inability to produce a toxin, and also deletion in genes in the biosynthetic pathways. So again, we have a similar story with a very strong but protracted domestication and with some diversification, but very recent, very strong selection. And selection for higher competitive ability, faster growth on cheese, change in color, loss of toxin production, and drastic loss in genetic diversity. And also, again, decrease in spore and loss of the fertility. So this time we really cannot induce sex in Penicillium camembert tea. And even the conidia, the asexual spore that we can buy here to inoculate and make camembert, they produce less and less of these spores. And there are a lot of actually fungi, mold that have been domesticated for cheese making, and all across the fungal tree. You can see some Scopulariopsis, Bifusarium, Sporandonema, and you have here the Penicillium. And interestingly, so you have here the Penicillium camemberti, and we also domesticated Geotrichum candidum, so a very, very distant genus, but it has almost the same phenotype on Petri dish as Penicillium camemberti, and actually we use it also for making camembert in mixture with the Penicillium camemberti because it remains white longer, and actually when you buy industrial white cheese with very fluffy and very white crust, usually it's more Geotrichum candidum than Penicillium camemberti, and so that's like a convergent selection of the same phenotype, the white and fluffy phenotype. And so with that, the conclusion is that so there have been a multiple independent adaptation events of Penicillium fungi to cheese, and with convergent phenotypic adaptation in terms of colors, loss of toxin production, fluffiness, ability to grow on cheese, loss of, and so there have been a rapid adaptation, and actually by the very same horizontal gene transfer, this starship. And with, again, in all cases, very strong bottlenecks, and then degeneration, loss of fertility, and even of asexual spore production. And we found that the PDO can either protect or threaten genetic diversity, because in the case of Penicillium roqueforti, it allowed to keep a bit of diversity, while in Penicillium camemberti, actually, so for PDO, you have to use this albino strain. So it actually maintained the loss of diversity. And so while we know since Darwin and Vavilov that we need diversity to adapt to strain improvement and so on, so that's a very high concern in mold and in all crops. So just to finish, we made a small animation movie of the story and domestication of this fungi. If you want to see, you have here the code, and I want to thank all the collaborators and students and you for your attention.
Thank you very much for this super interesting and nice talk. We don't have much time, but if someone has a quick question, you can also just raise your hand and I will look whether I see it on the participant list. So I don't see one for now, but I think you can also still type it into the chat and Tatjana can read it. And I mean, since we are already a tiny bit late, I would suggest that we just move on.
Sorry.
No, we are still perfectly in time, just not much time for questions. Yeah, so then I would suggest that we move on to the next speaker. This is Jia Zheng. So she's from Beijing University, and she will talk about the restoration of long-lived species. So please go ahead and share your screen.
Okay, thank you. Can you see my screen? Share it? Yeah. Okay.
Yes.
So I
Yes.
hope you
Yep.
can see it. Okay. But maybe I should move this part. I don't know. Okay, whatever. But it's a bit hiding. Oh, yeah, now it's good. Okay. Okay, so hello everyone. I'm Jia Zheng. I'm from Beijing Normal University in China. And I'm very happy to share with you one of my theoretical projects about how does inbreeding depression influence the restoration and reintroduction of long-lived species. So first, I will introduce inbreeding. Inbreeding is the breeding of close related individuals within a population, and this can normally increase the probability of offspring inheriting the same alleles from both parents. So like I show here in the picture, that the red leopard is actually an inbreeding individual because its parents are siblings. And inbreeding is normally a moral taboo in human society, but it is actually commonly distributed in animal kingdom. And there's a meta-analysis recently found that animal actually rarely avoids inbreeding. And if you want to estimate the level of inbreeding in a population, there are multiple ways. And the pedigree analysis is the most traditional way that you need to know the family tree of all of the individuals in population, and you can calculate the inbreeding coefficients by checking the relationship between individuals. And with the development of genetic and genomic technologies and the genetic marker analysis and population genetic models can also be used to analyze the inbreeding level. Okay, and what is the effect of inbreeding? It is commonly known that inbreeding can cause inbreeding depression for the population. So that means the reduced biological fitness in a given population due to inbreeding. It increased the likelihood of offspring inheriting deleterious recessive alleles from both parents. And inbreeding depression can lead to a decline in various fitness-related traits. For example, in the helmeted honey eater, the inbreeding have a long-lasting negative effects on the reproductive success throughout life. And in the blue tits, inbreeding can also reduce the recruitment probability. However, in other species like the New Zealand hihi and I think this is Hawaii crow, so the inbreeding in these two species found only influence on the early life fitness, which means the only influence on embryo development or hatching success. And I am very interested in the consequence of inbreeding on long-lived species. The reason is normally long-lived species has a long-lasting partnership. They can breed repeated breed for many years, and so this long terms of reproduction can buffer the negative effects from inbreeding on the population. So, but even so, there is some conflicting results appear over species. For example, in the killer whales and red deers, inbreeding were found limits the population from recovery. However, in the Mexico dolphins and white-tailed eagles, inbreeding does not affect the population restoration even when they experience very small bottleneck events. So, and further, from the dolphin study that they found, So they did the simulation here, and they found that if they increase the bycatch mortality, then the success of population restoration is also become lower. So that reminds me that maybe the various life history traits may interact with the effects of inbreeding on population demography. And this is especially important for the endangered species or the species that may go endangered in the future, to kind of predict what is the influence of inbreeding going to be on this population. And so here I raise a hypothesis that the stage-structured mortality rates and annual fecundity can collectively influence the effects of inbreeding on population restoration. And in this study, I mainly focus on the long-lived species with varied life history traits, long-lived birds. And the reason that I focus on avian system is because the long-lived birds normally have a long-term partnership, quite stable, and they produce a constant number of clutch size every year. And they also, in general, have monogamous system, and the mortality rate is acceptable for checking. And then I develop in video-based simulation to simulate the strides of inbreeding depression on the viability of bottleneck populations. And later we collect juvenile mortality rate, adult mortality rate, and clutch size data from 42 long-lived species in 15 families from a data set. And you can see this is a list of the species that we collect empirical data from different aspects. Okay, and then I will introduce my model structure. So first, we create a bottleneck population, include only 6 individuals, 3 males and 3 females, and we let them to randomly mating. And then once they get paired, they produce offspring. And then we will have a pedigree, which is a matrix for calculating the inbreeding coefficients. I will not go very detailed about this, how to calculate it, because it is a big part of the model, and it's very complex. So in general, the reason that we create this matrix is because we can check every individual's inbreeding coefficients by looking at their parents, grandparents, and the ancestors. And then the model will go to the reproductive stage. And here the clutch size is allowed to vary from 1 to 6x in different simulations. And the way that we determine the number of fledglings is by 2 models. So in the first version of the model, we do not involve the inbreeding effects to determine the number of fledglings. So the C is clutch size, and 0.9 is the hatching success we assumed. And in the second model that we introduced the inbreeding effects to this equation, and just to compare these 2 different the differences between these 2 models. And then we go to the mortality stage. So here we allow the juvenile mortality rate vary from 0 to 0.8. So you can imagine that every season we have a lot of new juveniles be produced. And so we will use the number of juveniles times the mortality rate, and then you will know how many juveniles will die in this season. And also we use the same way to calculate how many adults could die in this season. And then we remove all of the dead individuals from the population, and the adults who has lost their partner will return to the mating pool for finding a new partner. Okay, and then we let this process to run in a loop for many years until the population size recovered to over 5,000 individuals. And I need to mention that each individual can breed no more than 20 years in this model. And we run 100 simulations for each parameter sets, but we have a lot of parameter sets, which is about 960 sets. So this is really a huge amount of simulation. And then I will briefly show you the results. So this is the first model that model results that's showing without inbreeding effect. So here you can see that each panel is represents the clutch size from 1 to 6, and x-axis is the mortality of juvenile, and the y-axis is the mortality rate of adults. So here, the more green each grade is, means the higher chance the population can successfully recover. And if it's like the darker color, darker brown, means that the population could not restore at all. So you can also see some in some parameter zones, it has a median value. That means sometimes it can recover, sometimes it could not. And so here we can find that restoration probability really varies dramatically based on the combination of life history traits. And here I show the second model that's when we introduce inbreeding effects to this model. And you can really see that inbreeding depress a lot for the restoration success. Like if you compare to these two panels, you can see that the area, the brown area really become bigger. Okay. And what we did next is to mapping the species to the simulation outcome. So first I need to explain this figure. This figure is to use this figure, the value on each grade of this figure to minus that one. So we create a net effect of inbreeding depression. So the more orange this color it is, means the higher inbreeding will affect on the population restoration. And then we mapped all the species, the 42 species, according to their life history traits to this map, to this figure. And then here is the figure that's the original figure I showed in the last slides without inbreeding effects. So I will guide you a bit here. So we can see three scenarios. Like for example, for this species, it's barely got any influence from inbreeding. So you can see this color, the value is about 0.0, I think. So for this species, it has originally have a high chance to recover, and then the inbreeding do not affect a lot on this species. And for this species, inbreeding has a relatively high influence on it, because it's about 25% decreases on the restoration success. And for this species, it's become even more severe, because originally, if you do not consider inbreeding here, then the species still have a certain chance to successfully recover. But when we consider inbreeding here, this species has no chance to recover at all. So that means the inbreeding really have different extent of influence on different species. Okay, and then what we did later is we want to examine this theoretical framework with an empirical system. So here we find the crested ibis, which is very pretty birds you can see here. So why we select this species is because that the species has experienced a severe bottleneck events. So only 7 individuals has been reside in China, and in the past 4 decades, it's successfully recovered to over 9,000 individuals. And it's got well protected in national reserve, and they just raised this species. And nowadays, the ibis could already breed independently in the wild. So this is a successful example. So we want to bring the breeding parameters from crested ibis to our model, to tailor this model and to see how likely this species can actually recover from the theoretical way. And because the species got very good protection, so they have the full pedigree data, and they found that inbreeding only affects on the hatching success. And this is the relation between hatching success and inbreeding coefficients. And then we ran 300 simulations to estimate restoration probability, restoration time, and the population inbreeding coefficients. So here is the results. So first, I want to say that out of the 300 simulations, 90% of the simulations leads to a successful restoration, which is quite surprising to me. And in the A1 figure that you can see, the distribution of restoration time they spend for the population to recover is normally, well, is mostly gathered at a span 40, a bit more than 40 years to recover. And then the A2 is showing the population increases, how the population increases in size. And the A3 is very important actually. So here on the y-axis is showing the average inbreeding coefficients. So you can see that in the early simulation time, the inbreeding coefficients increase dramatically, and later it seems like it's reached to a platform in the successful restorations. And also the shadow, the light green shadows means the variation of inbreeding coefficients within the population. You can see that as the simulation goes by, that the variation become converged, more and more become smaller. Okay, and then later, so this is about, so if the simulation spent more time in restoration, then the final average inbreeding coefficients will also be higher. Okay, and then we compare the theoretical results with the empirical results. So this is empirical records. So the crest ibis spent 41 years to recover to 9,000 individuals. And from the simulations, we found 44 years, which is very close to this empirical time. And also the empirical inbreeding coefficients is about 1.165, which is also very close to our theoretical outcome, which is about 1.16. So we will say the simulation results match well with the empirical records. And then after this, we are considering a conservation program, which is about annual reintroduction. So what is a reintroduction? Reintroduction is a process of returning species to its native habitat after it has been extinct or nearly extinct in that area. And the strategy aims to re-establish a viable self-sustaining population in wild. Actually, reintroduction has been applied in many endangered species and also in different countries. And we also did that in chrysid ibis in China. And the reason that we do that is because there's a study, a simulation study found they predict the distribution of ibis in 40 years, around 40 years, and they found that the ibis has a low ability to expand the population by themselves because they are residential species. So then I want to ask, what reintroduction strategy should we use for endangered species having low ability for ability of population expansion? So then I create a follow-up model, a reintroduction model. So this model is aimed to evaluating population sustainability under different reintroducing strategies. So here I consider two types of reintroducing strategy. The first one, I call it the firework style reintroducing, which is because it looks like a firework. So the source population is the first restored population, and from this population, I will reintroduce some pairs, number of pairs to different locations, all from this one source population. And the second reintroduction strategy is called stepstone style reintroduction. So here you can see that what we did here is from the source population, we first reintroduce some individual to the first population, and then until it's recovered, and we reintroduce again from this population to the second population, and from the second population to the third population, and so on. So it's more like a sequential reintroduction, I would say. And so this is extension of the Christed model, Christed IBIS model, but it is actually aiming for providing a more generic conservation advice. And these two types of strategies are actually ongoing in a small scale in China. And so it is really demands theoretical evaluations for the efficiency of these two strategies. And here I'm showing the results of the simulation results of these two reintroduction strategy. So for the first firework style, you can see this is the result, and the different panel actually means how many pairs we plan to reintroduce to different populations. And x-axis is restoration time, and y-axis is the average inbreeding coefficients. And the green dots means a successful restoration, and the red dots means extinct, the population go extinction. And so you can see, even though when we introduce very few pairs to the new populations, there's a high chance that the population can still recover quite well. So in this 50 simulations, only one simulation go extinct. And if you introduce more pairs to different populations, then you will find that the restoration time becomes shorter. That means the population will develop faster. So this is the result from the firework style reintroduction. Okay, and in the scenario of stepstone style reintroduction, we found quite different results. So here still the different panel means the number of reintroduced pairs, and X and Y is still the same as previous slides. And I want to explain that the color of the dots means the rounds of reintroduction. So we run like 10 rounds in total. And the size of the dots means how many simulations can actually reach to this reintroduction rounds. So for example, when we introduce 3 pairs at each rounds, then you can see that the whole program can only reach to the 4th step. So there's no population could go further, and a lot of them just go extinct at the 3rd step already. But if you increase the number of pairs that you reintroduce to each population, then you see, for example, at the 9 pairs, if you introduce 9 pairs constantly, and then you can already see the reintroduction program can the full reintroduction program can works. And if you introduce even more pairs at each rounds, and you can see that the restoration time become much shorter. So for example, here in the 17 pairs scenario, that's you really see a much shorter restoration time compared to the 9 pair scenarios. But I need to remind that the inbreeding coefficient still increased quite a lot in this manner of reintroduction. So in general, I would say that the firework style reintroduction has a better conservation effects. So I will recommend that style to be the reintroduction strategy. So the take-home message here is inbreeding exerts various pressures on population restoration in species with diverse life history traits. And this model provides a good framework for identifying species that are particularly vulnerable to extinction due to bottleneck events. And the outcome of the tailored in-video-based model align with the conservation success of tri-state IBIS system. And lastly, the reintroducing individuals from the originally recovered population to multiple others, which is the firework style, can effectively ensure population sustainability. Okay, so I want to thank for my collaborators and the research fundings for this project. And I'm open for collaborations that request simulations for so many different aspects of ecological or evolution projects. And if you have system that you want to collaborate, just feel free to contact me. Thank you.
Thank you very much. That's super interesting and impressive how well your model fits with the data.
Yes. Thank you.
Yeah, does anyone have questions? So either type it in the chat or raise your hand. I do have one if no one else for the moment has, which is like, I mean, with this increasing inbreeding coefficient, could that pose a problem then at much later time scales? So when, I don't know, new environmental change happens or so, I mean, they would not be able maybe to react so well? Or what do you think about what may happen at longer time scales?
So you mean longer time scale in one individual's life or in a larger?
No, over longer evolutionary time scales. I mean, if you look longer into the future, I mean, like there might be so many environmental changes happening. I mean, is this reintroduction something that is long-term sustainable, or would you maybe see much later extinction just because they cannot, like, don't have enough genetic variation maybe to react to future environmental changes?
Yeah, I think that's a very good question. Although my model do not really focus on this subject,
Yeah.
but I think under the climate change, a lot of breeding biology information will also change accordingly. Like the clutch size may also change because the breeding window probably be shorter, and so a lot of birds actually breed more eggs in one go. So that will of course also influence on the results of our simulation framework. So it will definitely have effect in the long-term scale. But yeah, I don't know exactly.
Yeah, sure, it's not part of your model. I mean, yeah, I just wondered. Yeah.
Yeah. Okay, thank you. That's a good suggestion.
I think there was also a question by Franjo.
Oh
yes. But as the time does perhaps not allow it, I can perhaps discuss it at that time. But the point is that I wanted to make is that typically you have more severe inbreeding depression in populations that did not have a history of inbreeding. Because if you have several generations of inbreeding, then you get purging of negative alleles in the population, and inbreeding depression then goes down. So the effect of inbreeding will then go lower, will become lower. And I could imagine that if you take the stepping stone scenario, that then purging, so basically may be more efficient than the fireworks scenario. And therefore, although there is a higher inbreeding coefficient, inbreeding depression may actually go down.
Okay. Yeah, I didn't consider the purging scenario, but I know that there is some effects from that. So the effects from inbreeding is quite complex. It can affect a lot of different stage and also in different ways. So I can only say that here in this model, I only consider the inbreeding depression as a, yeah.
It's nice, but we can perhaps communicate about that.
Okay, nice to see you here. Thank you.
Okay, great. Yeah, thank you very much. And let's move on to our third speaker, which is Regina Borkom. And she is going to talk now about herbicide resistance. And yeah, I mean, we are moving from birds to plants now.
Hi, thanks everyone for being here. I assume everyone can hear me okay. I really appreciate Hildegard inviting me to give a talk in this symposium this morning. I'm going to give a brief overview of some of the work that we've been doing in the lab in the past 10 years or so, and then that will step us off into sort of our next group of questions. So in my lab, we've been working on herbicide adaptation in weedy plants in agroecosystems. And that's why my title is, it's fairly broad because we combine a number of disciplines. So it's herbicide adaptation as a model for integrating across genetics, ecology, and evolution. And so first, I want to start by just noting that plants live in a world that's dramatically altered by humans. There are many different ways in which humans have been altering the world. So whether it's the selective regimes from agriculture, or urbanization, or global climate change, plants are being exposed to really strong regimes of selection and having to either adapt to this selection, respond in a plastic manner, or else move their populations. And the focus of my lab for a while now has been on plants adapting to the agroecosystem. And specifically, the scenario that we work on is shown here with a tractor that's applying herbicide to a maize field. So effectively, a farmer can plant a crop that, let's say, is tolerant of a particular herbicide, most typically Roundup. The farmer lets the crop grow and then will spray herbicide across the entire thing. The crop survives, but then any sort of weedy plants that are existing in this field will die. And so unless, of course, they've adapted to the use of herbicide via resistance or tolerance mechanism. So the herbicide that is most typically used in the United States and worldwide with transgenic technology is glyphosate, which is the active ingredient in the herbicide Roundup, although we are beginning in the US to use an herbicide called dicamba, which we're also working on, but I'm not going to talk about that today. If you're interested, please feel free to reach out and ask a question. But the herbicide glyphosate, again, that's the active ingredient in Roundup. It's the most widely used herbicide, like I said, globally, as well as in the United States. And I'm showing you a map here of the estimated use of glyphosate on agricultural land in 2019. And you can see that it's in pretty heavy use in particular parts of the United States across a relatively large swath of land. But where you see the most intense use is in the Midwest, a little bit in California, and as well in the Southeast. And this brown area maps to where the majority of our row crops are. And so that's, our large crops of maize, soy, or cotton. And so with any sort of type of selection that we're imparting on really anything, but especially the agricultural ecosystem, plants will adapt. And I'm just showing you here an image of the increase in herbicide resistance over time. And so this is any resistant case in any weed to any herbicide, but you can see a dramatic increase of herbicide resistance over time. The work that we've been doing in the lab has focused on the evolution of glyphosate in the common morning glory, which is Ipomea purpurea. I'm showing you here an image of the species. The morning glory, you've probably seen it growing up trellises or on mailboxes. It's a very common, popular ornamental species because it has these really pretty big, showy flowers. The plant is a hermaphrodite, so it both selfs and outcrosses. It uses a mixed mating system to reproduce. You can see an image of it here right next to a soy field. The problem with the common morning glory is that once it gets into a disturbed area, it's really fantastic at taking over that disturbed area. And that makes it a really good organism to take over row crops, right? So you can see it vining over the soy. The problem with the species is that it will vine into the row crops, thus making it almost impossible in some cases for the farmers to get their combines in and harvest their seed. So it's typically considered one of the worst weeds, at least in the southeast of the United States. Some of the work that we have previously done is to assess the level of resistance to glyphosate in this species. We did this using a very large greenhouse experiment of about 10,000 individuals, where we went to different populations of the common morning glory and collected seeds, grew them up and sprayed them with the field dose of glyphosate. And what I'm showing you here on this map is populations coded in red. Those populations had a high survival rate. Populations that are coded in blue had a very low survival rate. So we have a couple of different hotspots of high resistance to glyphosate in the species in the United States. So what I'm showing is not only are there populations that have high resistance, there are also populations that are still highly susceptible and show low resistance. So there's still variability in natural populations for the level of glyphosate resistance. And one of the reasons that this might be so is that we've identified a fitness cost associated with glyphosate resistance in the common morning glory. So I'm showing you here some work from a previous postdoc in the lab named Megan Van Etten, where on the x-axis, I'm showing the proportion of resistance in the population, or the proportion resistance of each population. And on the y-axis, I'm showing you the proportion of germination, so the proportion of seeds that would germinate from that population. And you can see there's a very negative relationship between the two. So as the populations became more highly resistant to glyphosate, they also showed a pretty severe fitness decline in terms of lack of germination. So there's a fitness cost in this species, and the presence of a fitness cost plus row crop rotations might be one of the reasons that we're still seeing variability in nature for the level of glyphosate resistance. Now, more recently, we've sequenced the common morning glory genome and assessed the genome for signs of selection. And so what I'm showing you here is just a brief outcome of that. This is work from a previous grad student in the lab, Sonal Gupta. And I'm showing you the different chromosomes in this diagram, and then the little red spots and blue spots that you see on this graph are basically indicating areas of the genome that show signs of selection associated with glyphosate resistance. Now, we took a close look within those regions, and what we found are signs of selection on genes that are associated with transport within the plant system, as well as detoxification. So genes that are cytochrome P450s, glycosyltransferases, ABC transporters. So from this screen, we have a fairly good idea that the resistance mechanism in the species has something to do with altered translocation of the herbicide, as well as detoxification. Now, whether or not it's detox of the herbicide itself or the secondary compounds within the plant, we're still not sure. Okay, so these three pieces of information, whether or not there's variability in nature for the level of resistance, are there fitness costs associated with resistance, and what is the genetic or molecular mechanism underlying resistance? These are three really common themes, what you see in the literature of people who are assessing weedy plant adaptation to herbicide. So some of the work that we've also done is a little bit different. We've looked at the potential for traits that are correlated with the level of resistance in the common morning glory. And so what I'm showing you here is another piece of work from Megan Van Etten, where she's showing a PCA plot where we took germination traits, a bunch of different, are they germinating, are the seeds germinating traits, as well as early growth traits, so height, number of leaves. And so what I want to just point out here is that the red dots are indicating the level of Roundup or glyphosate resistance. So some populations that are highly resistant, say this population number 5 here, it shows effectively normal size compared to susceptible populations, but it was also showing really, really poor germination. So normal size, but poor germination. On the other hand, other resistant populations were showing normal germination compared to susceptible populations, but were really small in size. And so effectively what this is showing us is that as resistance evolves, there are particular traits that are correlated to resistance differently in different populations. We also found another type of trait that's correlated with glyphosate resistance in the common morning glory. we found that as populations increase their level of resistance, so I'm showing you here on the x-axis survival at slightly higher than the field dose of glyphosate, as populations became more highly resistant, they also showed a much lower outcrossing rate. So I said earlier, this species shows a mixed mating system across populations. It shows about 50% outcrossing. Some populations were highly outcrossing, up to 80% outcrossing. Other populations showed very low outcrossing, so highly selfing, about 20% outcrossing. And those populations that showed low outcrossing were highly herbicide resistant. So this is two examples of traits that are evolving or correlated with glyphosate resistance in the common morning glory. And we found these traits to be interesting because a weed plant, a weedy species, a population of a weed is existing in a matrix of a community, right? So there's other weedy plant species that they're interacting with. The plants are interacting with diseases, they're interacting with pollinators, and they're interacting with herbivores, to just name a few types of community interactions. So we began to wonder, does the evolution of herbicide resistance alter interactions between plants and other community members as a result of the evolution of resistance itself, or the evolution of these traits that seem to be correlated with resistance? And so this is sort of the next step of what my lab has been working on, and specifically thinking about herbivory. So we're asking, are there altered interactions associated with the evolution of glyphosate resistance? This is not the first time that someone has been interested in the concept of changed interactions with the community, given the evolution of resistance. So Sharon Strauss in 1999 in Evolution published a paper on a weedy plant, on Rhamnus. She took lineages of this radish and selected them for high resistance to flea beetles or low resistance to flea beetles. And then she looked to see if the plants were being pollinated equally. And so I'm showing you this graph. We've got the high resistance lines and the low resistance lines, and I'm showing you two bars for each. One of them just means that the plants were damaged. The other bar means the plants were undamaged, but that's not really the main point. The main point is that individuals from the low resistance lines had pollinators that spent more time foraging on them compared to the high resistance lines. And so this effectively shows that plants that have a higher level of resistance have different or altered interactions with a community member, in this example in terms of pollination. And so from this was termed the idea of ecological cost. So ecological cost is defined as a trade-off between fitness and resistance mediated from or through interactions with other organisms. Sharon Strauss and colleagues detailed what it means to show an ecological cost. And so basically you ask, one, does resistance alter interactions with other organisms? And two, do these altered interactions lead to changes in fitness? Okay, so that's how we would define an ecological cost. There's one example that I know of showing an ecological cost of herbicide resistance. And in this example, it's resistance to the herbicide triazine. Triazine is still a commonly used herbicide, typically used in maize crops. But Amaranthus hybridus, which is a really noxious weed, evolved triazine resistance. And work from Aaron Gaspin and Douglas Fatuma showed an ecological cost in this species. So I'm showing you two plots here, one of floral biomass, the other of vegetative biomass. And we've summarized it for biomass in terms of triazine susceptible lineages or triazine resistant lineages of Amaranthus. So effectively, what they found is that in the presence of competition in herbivores, lineages that were triazine resistant showed lower floral biomass and lower vegetative biomass than lineages that were susceptible. Okay, so this is evidence of a fitness cost. An ecological cost. We show a lowered component of fitness due to interactions with a community member and a resistant organism. So enter a grad student in my lab. This is Grace Zhang. She's a fourth-year PhD student. She wanted to understand if there is an ecological cost in the common morning glory. So is there some sort of a trade-off or negative relationship between herbicide resistance, in this case glyphosate resistance, and herbivory resistance? So to answer this question, Grace wanted to use the common morning glory system. And the reason that she wanted to was because we've already established some of the basic criteria for asking this question. So previously it's been shown that there's genetic variation for insect herbivory in the species. And as well, Ellen Sims' work in the late 1980s showed that there's positive selection for increased herbivory resistance. We've shown that there's genetic variation underlying glyphosate resistance in this species, and that there's positive selection in the field for glyphosate resistance. Okay, so we kind of have the basic criteria for the evolution of both glyphosate resistance and herbivory resistance present in this species. So Grace asked her question, are there ecological costs associated with glyphosate resistance in the common morning glory? By doing a common garden field experiment. So she took lineages from 18 different populations and 2 to 6 maternal lineages per population. She had replicates of each maternal line and each population for a total of about 1,600 plants that she planted in the field, and after about a month of growth, exposed half of the experiment to the field dose of glyphosate, which is 1.5 kilograms of active ingredient per hectare. She let the plants either survive or die, and then came and assessed damage. So she measured glyphosate damage as the percent of leaves that were showing damage. And so basically she would count the number of leaves on each individual, and then the number of leaves that were showing the telltale signs of glyphosate damage, which are yellowing and sort of a browning of the leaves. She also assessed herbivory damage, so the percent of tissue that was missing. She assessed 5 leaves per each individual. And from this, she estimated resistance to both glyphosate and herbivory as 1 minus the proportion or percent damage that the plant was showing. So we've got glyphosate resistance measured in this way, as well as herbivory resistance. So just as an aside, an interesting thing that she found is that herbivory increases when plants are treated with glyphosate. So I'm showing you here a common morning glory leaf from the control environment, where you can see some chewing herbivory there on the leaf. In the presence of herbicide, however, that herbivory was much greater. And when you look at this across each individual leaf from each individual plant, you can see that there's a higher proportion or higher percent herbivory damage per leaf in the treatment environment compared to the control. Now, Grace also assessed what kind of herbivory damage each leaf was showing. So for each of the 5 leaves, she said, is there whole damage, is there margin damage, or is there surface feeding damage? And from this, she assessed how did the patterns of different types of feeding change in the presence of herbicide. And so I'm showing you here on the y-axis the proportion of the leaves that were showing each type of damage. And we can see that in the presence of glyphosate in the treatment environment, there's more whole and margin damage. There's significantly more margin damage than there is whole damage. However, in the presence of glyphosate, there's lower surface feeding. So she found, not only did she find increased feeding of herbivores in the presence of glyphosate, she also found that the types of feeding changed. And so we don't have data to support this yet, but we're suggesting that perhaps the presence of glyphosate in a plant makes that plant unpalatable to particular herbivores, thus changing the patterns of feeding. Okay, so back to Grace's main question. Is there the presence of a trade-off between herbicide resistance and herbivory resistance, which could indicate an ecological cost in the common morning glory? Well, this is what she found. On the y-axis, I'm showing you the normalized glyphosate resistance and the normalized herbivory resistance, and we can see a strong positive relationship between the 2 traits. So this is pretty much totally opposite of our prediction of a trade-off between the 2 traits, and that we found a positive relationship between the 2. We also assessed, is there the presence of genetic variation for both types of resistance in the population that she was working with. We found the presence of genetic variation for herbicide resistance, but not for herbivory resistance. We also assessed, is there a link between both types of resistance and fitness? And what she found was that there is a link or relationship between herbicide resistance and fitness, as we've shown before. But in the study population, we found no link between herbivory resistance and fitness. So effectively, what this analyses or what these data are suggesting together is that given the presence of genetic variation for herbicide resistance and the link between herbicide resistance and fitness, in the presence of glyphosate, we would predict that glyphosate resistance could evolve to increase levels over time, and that herbivory resistance would probably hitchhike along with herbicide resistance, but by itself would not evolve. So why did we find this correlation between these two traits? Why did we find the exact opposite of what we were expecting? Well, this is a diagram showing how glyphosate works. So glyphosate impacts the shikimate acid pathway. This is the shikimate acid pathway. Glyphosate inhibits this EPSPS, which is 5-enolpyruvyl shikimate 3-phosphate synthase. It stops the production of EPSP synthase, which normally goes on to produce corismate. Corismate is a really important precursor to amino acids such as tyrosine, phenylalanine, tryptophan, and those amino acids are critical for producing important herbivory defense compounds such as alkaloids, glycosides, tannins, anthocyanins, flavonoids, glucosinolates. So what we're thinking is one model where here is the proposed mechanism of glyphosate resistance in the common morning glory. We have signs of selection on genes that are involved in translocation as well as detoxification, such as glycosyltransferases and cytochrome P450s. So in one sense, we think perhaps lineages that have the ability to detoxify or just resist herbicide are allowing this EPSP synthase or the shikimate acid pathway to go ahead and continue to produce corismate, which then produces the compounds that are necessary for herbivory resistance. And what I've just verbalized there would be sort of an epistatic interaction between the two resistance mechanisms, where you have to be herbicide resistant to be able to see the herbivory resistance. Another simple idea is that perhaps this is just due to pleiotropy. So we know that glycosyltransferases and cytochrome P450s are really important in herbicide detoxification, but they're also involved in herbivory resistance in some species. So we could have either an epistatic interaction going on, or we could have just simple pleiotropy between the mechanisms. To understand this, however, so the common morning glory is not a model system. I think it should be. And so to understand whether or not, or exactly how the two resistance mechanisms are working together, we need to be able to do functional genomics, right? So we can knock out particular genes and then assess how the different resistance traits are working. And so to do this, we're currently developing functional genomics in terms of developing magic mapping populations, fast neutron mutant lines, and CRISPR-Cas transformation protocols. So we're in the middle of developing these resources for the Morning Glory community and plant science community. So if anyone is interested in working on Morning Glories, please contact us. We would be very happy to have you involved. Okay, so to summarize my talk, we were wondering if is there any evidence for ecological cost associated with glyphosate resistance in the common morning glory. And here I'm showing you a population that has both herbicide resistant and herbicide susceptible individuals. If we were predicting a trade-off between herbicide and herbivory resistance, we would see something more akin to what Gassman and Fatuma saw, where the lineages that were herbicide resistant showed more herbivory. However, we found the opposite. We found that herbicide and herbivory resistance are positively correlated. showing that individuals that are less glyphosate resistant showed more damage from herbivory. And what this effectively would mean over time is as the population becomes more glyphosate resistant, it should also become more herbivore resistant, which could change interactions with the herbivore community and leading to changes of which we couldn't predict. Okay, so that is the end of my talk. I would like to thank all of these people that have helped me in the last 20 years or so, and I've highlighted different pieces of work from each of the people that I'm showing you here, as well as showing my funding sources, and I'd be happy to take any questions.
Thank you very much for this talk. Yeah, are there any questions? Maybe raise your hand or put it into the chat. I don't see a raised hand for the moment. There is, I see one. Yeah, okay. One.
Hello. I was wondering about what you know about the nature of the resistance and the dominance. So could the association with selfing be because it's exposed in homozygotes where it develops faster with selfing?
So yeah, so typically herbicide resistance mutations are dominant within the population. So I'm not really sure. Can you repeat your question?
So I was just wondering, the association with selfing, could that be because it's more exposed, the resistance mutation? So if it was recessive, you might see it might be developed faster. I know it's been dominant in a lot of other things, but has it been tested in the species, the nature of the inheritance?
So the nature of the inheritance. So we, well, with the common morning glory, we think it's, polygenic, so multiple genes acting together. And we have some preliminary data suggesting that, yes, it is dominant and not resistance mutation. So we know that the ability to self in the common morning glory is typically due to anthostigma distance. And we know that anthostigma distance is typically a single gene, right? And so what we're predicting with the increase of those two traits together is that it's a reproductive assurance sort of argument where individuals that are resistant don't need anyone else around necessarily to cross with. And so that's why you would have the correlation between those two traits.
Okay, thank you.
Thank you.
I have one question. I mean, it's a very naive one, but in this herbivore that you were looking at, is it something that is then also found in the field and attacking the really crop plants? Or because in that case, one may also then use whatever insecticides or so against it. So I wondered, is it something that is also happening in the field, or would this herbivore rather be somewhere in nature, not in the crop field itself?
Yeah, that's a great question. So most of the herbivores that we saw eating morning glories were Japanese beetles. And so Japanese beetles will just eat most anything. And they're kind of everywhere. So.
Oh, okay, I see. Okay. And is it, do you know whether then there is simultaneous application of insecticides and herbicides in the field?
Yeah, that's a great question. I think it varies farmer by farmer, and that's not something that we've looked into yet. But I do think it's an interesting question because sometimes insecticides can influence plants to turn on their other resistance machinery or their just general resistance machinery, and those two types of application of pesticides could interact and work interestingly together. So,
Thanks. Okay, so if there are no more questions, then the first part of the symposium is over. There's a coffee break now of 30 minutes, but really, I mean, thank you to all the speakers. I really enjoyed all three talks very much. They were really great, very very inspiring, very nice. And yeah, so I hope to see everyone back after the coffee break in 30 minutes. Hello.
Hello again.
So I will only give a very short, brief introduction, not to take up much time.
Perfect.
So that you're not surprised.
That sounds great. Let me make sure that I can still share. Yes, great.
Good.
I got myself more coffee.
Yeah, I admire that you are getting up that early.
I prefer early to late, so it worked out well.
Mm.
Where in Germany are you based?
In the very north, so it's not that far away from Denmark.
Oh, lovely. Is your summer in full swing?
At the moment, it's really nice weather. Yeah, I mean, northern Germany is not famous for, it's sunny, but at the moment, really, it's good.
Yeah.
It's a very nice region here, so plenty of lakes and
Yeah.
plenty of birds.
Yeah.
I enjoy that.
I've only been to the south of Germany, so one day I will get there and see more of the country.
Yeah, you should come up north.
I hear it's beautiful.
So where are you going in the south? Which?
I spent some time in Stuttgart and München. I had a friend in high school who grew up near Stuttgart. So
Okay, I see. Yeah. Yeah, I'm originally from the south, so.
Okay.
Let's maybe still wait a minute or so because I think there are probably a few more people.
Of course.
Sorry, I'm going to check on something. I think we might be in the wrong session because there are two. Well,
Oh, so I mean, we just stayed where we are.
oh yeah, so you should leave and then go
Oh.
into the next time slot. So they're probably waiting for you there. Okay.
Oh, okay. Thank you.
Yeah, sure thing.